There you go. So hello, today we are back with another mini lecture, and I am Millennium, Isekai'd Mask Knight Empress Scientist. So let's start with carbohydrates, its structure and metabolism. All right. So let's go with carbohydrates are essential biomolecules, and they play a crucial role in energy metabolism. They're classified based on their functional groups and could be simple sugars or polyhydroxyketones and aldehydes. So the aldehydes and ketones, those would have the magical C double bond H, C double bond O, I mean. So a simple aldehyde like propanol is like CH3, CH3, CHO. The CHO ketone also has it, but instead of a CHO, it's a C double bond O. And the ending is like it has a mini methyl at the end. And then you have an aldotriose also composed of aldehyde and ketone. An aldehyde and a ketone. You just have to make sure you know the differences between an aldehyde and a ketone. Aldehyde C double bond O and an H at the end. Meanwhile, ketone is C double bond O and a CH3 perhaps. Next, you have monosaccharides, and yeah, like those are the simplest form of carbohydrates, and they can be classified by the number of carbon atoms in their structure. Okay, and hexoses, sugars with 6 carbon atoms, are common monosaccharides, and the two primary examples are glucose and fructose. Glucose is an aldohexose. It has an aldehyde at the carbon 1 position. So again, glucose has an aldehyde at the carbon 1 position, and it's a very key source for energy in the body. And also, the OH on carbon 1 can be above or below the plane of the ring. And you have alpha glucose and beta glucose. Wow. And the alpha and beta forms are anomers, and they differ there in the position of the carbon 1. Fructose, on the other hand, is a ketohexose. It is a 6 carbon sugar with a ketone group at the carbon 2 position. And that's what makes it into ketose instead of an aldose. It also forms a ring structure, and the structure of the alpha fructose is similar with the alpha glucose, except for the position of our dear functional group. Galactose differs in the orientation of the OH group because the OH is on carbon 4. It's also an aldohexose and when it forms a ring, it's similar to glucose. It's essential in the body and especially in the synthesis of lactose. So energy metabolism in humans it has two primary goals. One is to obtain energy in the form of ATP and also to maintain a more or less stable or constant level of blood glucose for the cells, especially for the brain and CNS, which require it. And during starvation, you would, like, for example, under conditions of fasting or very low carbohydrate diets, the energy will be derived from stored triglycerides, TAG. And the blood glucose is primarily maintained, it's maintained by glycogenolysis and gluconeogenesis. So those are primarily from protein. So again, energy is provided primarily by stored tags if you are starving. And so the main question would be, how does the body prevent pyruvate from glucose or other sources from being used for energy? and thereby preventing the use in other metabolic processes. So hopefully we get to answer that later. Later. So living systems would derive energy from oxidation reduction reactions. And wait one moment. Let's go with, there you go. Yeah. So living systems derive energy from oxidation reduction reactions, which are fundamental biological processes. And these reactions would help convert glucose and other nutrients into energy. And with that, you can generate ATP to power the cells, especially like as a byproduct of these reactions. The system will then release CO2 and water. So again, you have food coming in and oxygen coming in. That's, you are a human, hopefully. So you have food and oxygen coming in, and then you have CO2 and water coming out. Hopefully, those are coming out. And then you also have energy coming out as well. And then, so let's go and talk about oxidation state. So the oxidation state of a carbon atom refers to the number of bonds it forms with oxygen minus the number of bonds it will form with hydrogen. So it's like oxygen minus hydrogen. The bonds. We're talking about bonds, okay? So with each bond to oxygen, it's a +1, and with each bond to hydrogen, -1. So oxygen +1, hydrogen -1. For example, you have CH3, CH2OH, and the central carbon, the middle one, would be a -1. So if you draw that out, you have the H2 in the middle, CH up, CH down, and OH at the right, CH3 left. So it says every bond with the C to the O is a +1. You have a +1, C to H -1, C to H -1. So we have a -1, -1 +1. What is that? It's a -2 +1. You have a -1 overall. Yay! And then how would you, what's the nomenclature of oxidative enzymes? Well. You have, enzymes involved in oxidation reactions are based on their specific actions. For example, oxidases would oxidize. It catalyzes oxidation reactions, while dehydrogenases are involved in the removal of hydrogen atoms from the molecules, and these enzymes are categorized based on their function, such as the classification of oxidative enzymes that interact with substrates to facilitate the breakdown of it. So again, a formal form would be oxidoreductase, but the general form is dehydrogenase. And if it's oxidation by molecular oxygen, it is called an oxidase. If it's by incorporation of molecular oxygen, it's an oxygenase. And if it's reduction, it's a reductase. Just terms depending on its function. So again, oxidation state, if it's a bond to oxygen, it's +1, bond to hydrogen -1. Carbon to carbon bond would be 0. So generally oxygenation refers to the addition of oxygen atoms or the removal of hydrogen atoms from a molecule. So for example, an alcohol has a higher oxidation state than a simple hydrocarbon. This is due to the additional hydroxyl group, which makes the molecule more oxidized. So it's just think of it as, okay, it can be more oxidized because of it. That's a leader. So here we have a table of common oxidation states, and it shows a couple of organic molecules. we have the methane, CH4. It has the carbon oxidation state of -4. Carbon dioxide, CO2, +4. This would, because it's CO, right? 4 bonds, C, carbon dioxide, CO2. C, and then O and O. C double bond O, C double bond O. So how many bonds? It's a +1, +1, +1, +1. That's why it's +4. Meanwhile, methane, CH4, CH. That's 4 H's. So it's -4. Bang, bang, bang. Okay, next, metabolism overview. So metabolism is a series of biochemical reactions that convert food or glucose and oxygen into energy. And this process occurs through oxidation reactions involving the redox and coenzymes, primarily the NAD plus and NADH. So with that one, glucose obtained from food is oxidized in multiple ways, okay? Multiple ways. So for example, the basic equation would be, let me just write it out. It's food or glucose plus oxygen equals ATP for the energy. So food plus oxygen go into ATP. So if you draw that out, I'm still figuring out how to draw over here. You have a big box. At the top of the box, you have food coming in, that's glucose, and oxygen going in also. And then in the middle, you have a cycle of from NAD plus going into NADH and going back. And then you have CO2 going out, H2O going in, ATP is also going out. So yeah, the oxidation of food is carried out by redox coenzymes, and NADH is now the reducing one. And the reduced coenzymes are reoxidized by oxygen. So that's why it's just a mini cycle. And the first step in metabolism of glucose is glycolysis, which occurs in the cytoplasm. And glycolysis breaks down a molecule of glucose into 2 molecules of pyruvic acid, a 3-carbon compound. And during this process, a small amount of ATP is generated. NAD plus, the oxidizing agent, is reduced to NADH. And this reduced form would carry the electrons to the later stages of metabolism. So again, the first step in metabolism of glucose is glycolysis, and it is in the cytoplasm. One more time. What's the first step in glycolysis in the metabolism of glucose? It is glycolysis, and it occurs in the cytoplasm. Glycolysis breaks down a molecule of glucose into 2 molecules of pyruvic acid. Glycolysis breaks down 2 molecules of glucose into pyruvic acid. During this process, a little bit of ATP is generated. NAD plus is the oxidizing agent. It's now reduced into to NADH. And this reduced form of NADH carries electrons to the later stages. All right. And now you have pyruvic acid into acetyl-CoA. And that's very important because this reaction is in the mitochondria. So, just to remind you, you have glycolysis, which occurs in the cytoplasm, but the TCA cycle or Krebs cycle happens in the mitochondria. Pyruvic acid is oxidized into acetyl-CoA. Acetyl-CoA enters the Krebs cycle, aka also known as the citric acid, and this is a critical form that bridges the anaerobic glycolysis with the aerobic pathway, which relies on oxygen. So you have glucose in the cytoplasm. It gets converted into pyruvic acid, a 3-carbon compound. And then with that one, hello Lendor, welcome back. Yes, another day, another adventure with science. And so we are discussing glycolysis right now, and it's the first step in the metabolism of glucose, and it happens in the cytoplasm. It would break down glucose after a couple of steps into pyruvic acid, and after that, pyruvic acid is then converted into acetyl-CoA. And how does that happen? That happens in the TCA or Krebs or citric acid cycle. And that's a very important thing because it happens in the mitochondria and it bridges the anaerobic glycolysis with the aerobic metabolic pathway, which relies on oxygen. Krebs cycle would release carbon dioxide as a byproduct. And also, you have NADH and FADH being produced. Important things for the electron transport chain. The NAD plus would be the oxidizing agent, while the NADH later on becomes the reducing agent. So yes, that is where you actually gain a ton of NADH and FADH too. And the final step is the ETC or electron transport chain. It occurs in the inner mitochondrial membrane, and the NADH generated from both glycolysis and Krebs would carry high energy electrons to the ATC. Why is that important? Because that's where oxygen plays a role as the vital or final electron acceptor, and it would combine the electrons and protons to perhaps form water. Also, the movement of electrons through the ETC would create a proton gradient across the mitochondrial membrane, and it would drive the production of ATP via oxidative phosphorylation. And this process would ultimately result in the production of bulk ATP used by the cell for energy. All right. Next, for the outcome, okay? In summary, the food or glucose is fully oxidized into CO2 and water, and you have a release of ATP. ATP would be the energy currency for the cell. You have the anaerobic reactions, glycolysis, and aerobic reactions such as Krebs, and also the electron transport chain. And the key takeaway would be, in the presence of oxygen, the key takeaway is that glucose, in the presence of oxygen, it's broken down step by step into smaller molecules in the form of ATP. So far it's true, and it becomes ATP. It's like glucose, acetyl-CoA, CO2, but there's like some special stuff going on. For example, in certain cells, like red blood cell or muscle cells during exercise, oxygen may not be available. So what do you do? In this step, the pyruvic acid would be converted into lactic acid instead of acetyl-CoA. Like, oh no, why? Because the lactic acid would still help in the regeneration of NAD plus and NADH, even if you don't have oxygen. But in the process, NADH like acts as a reducing agent, and lactic acid is the end product. So it's like, okay, you can still make NADH and like the others, but why is that bad? Because lactic acid buildup is not great. So we can talk about that again later. So for now, the two major fuels would be glucose and acetyl-CoA. Glucose, again, would undergo glycolysis, breaks down into pyruvic acid, produces some ATP. Pyruvic acid becomes acetyl-CoA, goes inside the Krebs. Acetyl-CoA is further oxidized into numerous stuff to produce more NADH, and you also have some FADH2 being made. The electrons go to the electron transport chain. But here's the thing. While glucose is the primary fuel used by the body for ATP production, other foods like fat and protein also contribute because fats can break down into acetyl-CoA. And it can also enter the Krebs. Proteins also can be broken down into amino acid, and some of those amino acids can be converted into glucose or acetyl-CoA, which will then fuel the other stuff. But note, big note, while fat can be converted into acetyl-CoA, it cannot be converted into glucose directly. So that's a key distinction between, like, when you compare glucose and fat as a source, okay? There's also the thing with ketone bodies. So what are ketone bodies? Well, when the body runs low on glucose, especially during extended fasting or carbohydrate restriction, ketone bodies are produced as a byproduct of the fat metabolism. These can serve as like an alternative fuel for the brain because they're also made from acetyl-CoA. So but that's just like an emergency procedure. Preferably don't do it a lot of times. Okay, next, final thought for that section would be, while glucose, acetyl-CoA, and fat all serve as essential energy fuels or sources, not all food can be converted into glucose. And that's why you have gluconeogenesis, which allows the creation of glucose from other substances. Fat cannot be converted directly into glucose. So this mini summary, energy production in the body is very versatile, relying on carbohydrates, proteins, and fats, and ultimately they would feed into glycolysis or acetyl-CoA production to produce ATP, with each contributing differently depending on availability. So they would just switch. If you're not available, they go use the other ones that are there. The mini review of bioenergetics. They involve the study of energy flow through living organisms, and that would focus on how cells convert energy and maintain metabolic functions. A key concept is the importance of NAD plus and NADH in cellular processes. We have ethanol metabolism. I think we talked about this a little bit earlier, but let's review. Ethanol is metabolized in the liver by a series of enzyme catalyzed reactions, and these reactions involve the conversion of ethanol to acetaldehyde. Again, ethanol to acetaldehyde by what? Alcohol dehydrogenase. The acetaldehyde would then be converted to acetic acid, and that's used in the TCA cycle to produce ATP, because acetic acid can be converted into acetyl-CoA. Very, very good detail to remember. Acetic acid can be converted into acetyl-CoA. So yes, you have ethanol to acetaldehyde using ADH, and that would also lead to a reduction of NAD plus 2 NADH. You have acetaldehyde dehydrogenase, which would convert the acetaldehyde to acetic acid. Again, use it from NAD plus, you have NADH being made. And then the acetic acid is made into acetyl-CoA, enters TCA to produce ATP. But here's the catch. The acetaldehyde is very, very, very toxic to the body. I don't think you want toxic stuff inside your body. And that would make the rapid conversion to acetic acid very essential to reduce the harm. Acetaldehyde accumulation can lead to hangovers such as nausea and vomiting. Note this down. Alcohol dehydrogenase is quite specific. So, or low. It's not specific. So alcohol dehydrogenase has low specificity. Meanwhile, it can metabolize other alcohols as well, such as methanol. And if you metabolize methanol, you can make formaldehyde. That's bad. That is bad. You do not want that to happen. That's toxic. Okay? So, in a similar manner, we talked about ethanol, alcohol dehydrogenase into acetaldehyde. Kind of the same pathway. If you have methanol, it can be converted to formaldehyde, and then formic acid. It's really not good. And you can treat that with fomepizole. It is an alcohol dehydrogenase inhibitor. Or you just add ethanol to compete with the methanol for metabolism. So you prevent the toxic buildup of the formic acid. Yeah, just don't drink methanol. All right. I think we talked about that one. So for energy reaction, you have it's glucose and oxygen reaction. You have the oxidation of glucose to carbon dioxide and water. Too big. Okay. It releases 686 kcal per mole of energy, and this energy is released in reactions like cellular respiration, which the body uses to produce ATP. Okay, you have some specific values here, such as carbohydrates would need 4 kcal, proteins 4 kcal per g, also fats is 9 kcal per g, ethanol is 7 kcal per g. Note, we have Gibbs free energy. Just a mini tidbit. If Gibbs is negative, it's exergonic. Okay? Exergonic means it will just happen. Reaction is spontaneous. And meanwhile, if Gibbs is positive, G plus, it needs energy input. It's non-spontaneous. G equals 0. Reaction is in equilibrium. No net energy is gained or lost. So G, just remember, G minus, it will go. You have enthalpy, or change in enthalpy, that's the H. So T is temperature, change of S is change in entropy. Entropy is like the disorder, and the maximum work is spontaneity. So just remember the formula: G, change in H, G equals H minus TS. Just add the triangles. Okay. Yeah, for a reversible reaction, you would need to apply work equal to change of G to move the boulder, if you think about it in a boulder way. And then you have Gibbs free energy. That was it. You have the standard Gibbs free energy, the G with a mini circle at the top. The formula for it is change in G equals G circle plus RT. Again, change in G equals G circle plus RT ln B over A. Technically, it's products over reactants. I think another one would be change in G circle equals negative RT ln KEQ with KEQ being the equilibrium constant. So it's just two versions of the formula depending on what you're looking at. If you want the G or the GO, the standard version of G, just note that down. G and G. Oh, when this next. So yeah, acetaldehyde is toxic, and that's why you want to convert it quickly. Methanol poisoning involves forming formaldehyde and formic acid, and you have bioenergetics involving the change in G, energy reaction for glucose oxidation to CO2 and H2O is 686 kcal per mole. And so we have a question. What is the parameter that is equal to 0 when the reaction is at equilibrium? Is it change in G or the standard G? It is the change in G because that's the definition of equilibrium. Next. What depends on the concentration of reactants and products? It would be change in G because the standard version is constant. So it's just the normal G. Next, if you want to practice calculating, you can do that, but just remember the two formulas so far. It is the G equals GO plus G equals GO plus RTLN P over R, or G standard equals negative RTLN KEQ, with KEQ being products over reactants. Bam bam. Bling. Next. Okay, ATP. ATP plays a central role in energy transfer within biological systems. When ATP is hydrolyzed, you get energy, which drives other stuff. Just use it up. So you also have coupled reactions because in biological systems, unfavorable reactions, aka those that require energy, can be driven by coupling them with favorable reactions. And it's important in the case of ATP hydrolysis, which is commonly coupled with reactions that require energy to proceed. So, for example, you have the hexokinase reaction. It's glucose plus ATP and the glucose 6P plus PI. So that, and hexokinase is, I think that's the thing that is used. It's the enzyme that will lead to it. Membrane transport and change in the Gibbs energy. Well, in systems where substances move across a membrane, change in G depends on the concentration gradient. So if the concentrations are equal on both sides, change in G is equals to 0 because it's at equilibrium. If there is a difference in concentration, change in G is not 0, and energy will be released or required depending on the direction. So if you want to note for membrane transport, the equation is earlier. Change in G equals change in G 0 GO plus RTLN B or RTLN products over reactants. So next is free energy for phosphorylation reactions. You have some esters such as phosphoryl esters, thioesters, and phosphoenol pyruvate. Phosphoryl esters would just be the standard would be negative 7.3. Thioesters is negative 9 kcal per mole, and then PEP, phosphoenol pyruvate, would be change in G, the standard free energy equals, it is negative 11 kcal per mole. So just remember that the standard free energy change under standard conditions is the G with the with this mini circle at the top. I don't know what it's called. I think it has a name. Change in G is the actual free energy change. It varies depending on the concentrations with reactants and products. The normal G, coupled reactions would allow unfavorable reactions to proceed by coupling them with energy releasing processes like ATP hydrolysis. And so that's why if a thing is unfavorable, most likely it would need ATP. Membrane transport involves changes in the Gibbs free energy, and that's driven by concentration agents, and that allows energy to be harnessed for moving substances across the membranes. Okay, and then I think, let's see. Yeah, I have more for carbohydrates over here. But I think we need to drink water, bro. Water break. 3 seconds. Let me just grab the tubing. I'm back. So for carbohydrates. Okay. Why is 1 second, I'll drink water. Okay. So why is ATP hydrolysis energetically favorable? And how does this drive some otherwise unfavorable reactions in glycolysis? That's true. It's so ample. It's over there a lot of times, you know. So ATP hydrolysis is favorable due to the relief of electrostatic repulsion. What is that? Well, it has increased resonance or stabilization of the pi and some solvation effects. What does that mean? Just note, if you break it up, much better. It feels better. It nets it. And yeah, what distinguishes ATP to ADP plus BI versus the other stuff? The first breakdown is negative 7.3. That's from ATP to ADP. But ATP to AMP releases a specific amount. It's, you know, negative 7.3, negative 7.3. That's negative 14.6. How would I design an experiment using radio-labeled ATP to determine whether a given reaction involves direct ATP hydrolysis? I wonder how. So, I can use a gamma 32 or that's the P, I think that's ATP. And you can run the reaction and track the radio label. So you just track the label. If the 32P appears in the products, ATP hydrolysis occurred. If not, ATP may be acting allosterically or it is in a transfer reaction. So how would I set up like a reaction? How to detect coupling between ATP hydrolysis and glucose phosphorylation in vitro? So how would I do that? Good question. I can set up a reaction with hexokinase, glucose and ATP to measure and I can measure the products of G6P, by the NAD plus and G6PDH assay. And if ATP disappears, maybe the stoic conversion would confirm the coupling. Okay. So if we want to talk about energy investment and payoff phases of glycolysis, so we have the investment phase, that's the hexokinase and PFK1. Cleavage would be aldolase and triose. And then you have the payoff phase later on with GAPDH, PGK, enolase, endolase, and pyruvate kinase. Why are hexokinase, PFK1, and pyruvate kinase considered irreversible? Their change of G standard is very negative. So that means they It wants to happen. So it's very unidirectional. And that's why they act as metabolic valves. How is PFK1 regulated allosterically and hormonally? And why is it a powerful activator? Well, hexokinase has a low Km and low Vmax, and meanwhile, glucokinase has high Km and high Vmax. So it just depends on that one. Chun, chun, chun. Okay. What determines the direction of electron flow in the ATC? It flows from redox pairs with a more negative E. And why does FADH2 yield less ATP than NADH? Because FADH2 would enter at complex 2. It would bypass the complex 1, which has no proton pumping. And that's why fewer protons are translocated. That's why FADH2 is around 1.5 versus NADH would yield around 2.5 ATP. Okay, so those are very specific details for that one. And now we move on to a little bit of glycolysis. So ATP is a central source of free energy in the cells, breaking the bonds between the beta and the gamma phosphates of ATP would form ADP and inorganic phosphate. And many biochemical reactions are not favorable because of their high positive G. For example, formation of G6P from glucose and PI, it has a plus 3.3 kcal per mole, and it's not really spontaneous. That's why you couple it with ATP hydrolysis, which can drive it forward. Okay, sticky note that. And certain compounds have high negative free energy of hydrolysis and are considered high energy molecules. These would include phosphoenol pyruvate. So it has a negative 14.8, so PEP would really want to go bam. And then the 1,3-bis phosphoglycerate has a negative 11.8 kcal per mole energy. Thioesters like acetyl-CoA also has negative 7.5 kcal per mole. So their hydrolysis can be used to synthesize ATP. That's why acetyl-CoA is very important. Coupled reactions. Oh yeah. Next, glycolysis. Let's talk about this in greater detail. Okay, glycolysis. It begins with glucose. It is a 6-carbon molecule, and it gets converted into 2 molecules of pyruvic acid. And this occurs in the cytoplasm. It does not require oxygen. ATP is initially consumed in 2 phosphorylation steps. Okay, so you have glucose into G6P, and then again make the fructose 6-phosphate. So that's where, when fructose 6-phosphate is converted into fructose 1,6-bisphosphate, that's where you use up ATP first. So you have glucose, use up ATP to convert it into what? Glucose 6-phosphate. Glucose 6-phosphate converted into what? Fructose 6-phosphate. Fructose 6-phosphate into fructose 1,6-bisphosphate. And you use ATP there. And then after that, that final 6-carbon sugar is split into 3 carbon molecules, glyceraldehyde 3-phosphate G3P, and dihydroxy acetone phosphate. All right, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Dihydroxyacetone phosphate, you can convert it into G3P. Well, we want to convert it to G3P. Why? Because G3P is then converted into 1,3-bisphosphate. Bisphosphoglycerate. The conversion of G3P into 1,3-bisphosphoglycerate would yield NADH. NADH is good. That's good. We will use that later on. Okay? And that is substrate level. After that one, 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. And when the phosphoenol pyruvate PEP is converted into pyruvate. Yeah, after that one. So right now we have, let's start again, glucose into glucose 6-phosphate. Then glucose 1,6-bisphosphate. You break the nope, never mind. Again, glucose. You have, and then with using ATP, you have glucose 6-phosphate. After that, fructose 6-phosphate, then fructose 1,6-bisphosphate. Break that up into G3P and DHAP. DHAP converts into G3P. G3P would then yield NADH if you convert it into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate into 3-phosphoglycerate to 2-phosphoglycerate. You have water yielding out, and then you have PEP. And then ATP again. So there's a lot of terms over there. I didn't even mention the, what's that called, enzymes yet. But that's overall what happens without the enzymes. For now. We need the enzymes though. So slowly but surely, then we're gonna go there. So glucose, again, a 6-carbon sugar is phosphorylated using 2 ATP to form fructose 1,6-bisphosphate, and like with other steps, and that is then split into DHAP and glyceraldehyde 3-phosphate. DHAP would then be converted into G3P, HAP, and at this point each reaction happens twice because you have it happens twice per glucose molecule because you broke it up. So the net result overall of glycolysis is 2 ATP is used in the investment phase, 4 ATP is made in the payoff phase, so that's why the net gain is 2 ATP. 2 NADH is made and 2 pyruvate is formed. Alright. Glycolysis, we talked about this part. Yeah, and the specific, and it's very, very specific steps. But this one has the enzymes. Okay. For the enzymes, we have, let's start. So we have glucose, we have hexokinase. Go into glucose 6-phosphate. And then with that one, you have phosphoglucose isomerase, the enzyme, into fructose 6-phosphate. So glucose, using hexokinase, into glucose 6-phosphate. And then you have phosphoglucose isomerase because it just switches the form into a fructose, so you have fructose 6-phosphate. Phosphofructokinase would make it into fructose 1,6-bisphosphate. Then you have into DHAP and G3P. Aldolase breaks it up. We mentioned earlier, from DHAP into G3P, what makes it? You have triose phosphate isomerase. From G3P, you have glyceraldehyde phosphate dehydrogenase. It's glyceraldehyde phosphate, glyceraldehyde 3-phosphate. So that should be easier to remember. Hopefully. And then after that, you have 1,3-bisphosphoglycerate. You have phosphoglycerate kinase for that one. And 3-phosphoglycerate. Bam, bam, bam. Later on, PEP, pyruvate kinase to pyruvate. That's the important part. So just remember, pyruvate is the ending. And then you have gluconeogenesis. What is gluconeogenesis? It's just the reverse form, the opposite version of glycolysis, because glycolysis breaks down glucose to form energy, to produce energy, while gluconeogenesis synthesizes glucose from non-carbohydrate precursors. However, because several steps in glycolysis are thermodynamically irreversible, like gluconeogenesis would use different enzymes to bypass those steps. So for example, the three irreversible steps in glycolysis were like the hexokinase converting glucose to G6P using ATP, and that is bypassed by gluconeogenesis using glucose 6 phosphatase. It would remove the phosphate group without using ATP. And then the second irreversible step in glycolysis was phosphofructokinase PFK1 phosphorylating the F6P to F16P BP. In gluconeogenesis, fructose 1,6-bisphosphate is used. Phosphatase, it hydrolyzes the phosphate group. And then the third irreversible step in glycolysis was pyruvate kinase. It converts the phosphoenol pyruvate to pyruvate while generating ATP. And gluconeogenesis bypasses this using a two-step reaction. It would use pyruvate carboxylase, converting pyruvate to oxaloacetate using ATP, and PP carboxylate would convert to oxaloacetate to PP using the GTP. So there's a lot of things going on. And you have to remember that. Aldolase, we talked about how it cleaves fructose 1,6-bisphosphate into DAP and G3P. It is an aldol cleavage, reverse of an aldol addition. Never knew I need. The mitochondria is the powerhouse of the cell. Let's go. Of course, we have to remember that the mitochondria is indeed the powerhouse of a cell. And good job with the timing, JPEG, because we're talking about glycolysis and Krebs. Good job. So right now we are at the aldolase summary section. Aldolase would cleave fructose 1,6-bisphosphate into 3 carbon intermediates, and that's the dihydroxyacetone phosphate, the DAP, and G3P. This reaction would split the 6 carbon sugar into half. 3 plus 3 equals 6. So that's why. Carbons 1 to 3 of the original glucose molecule would form DAP, the dihydroxyacetone phosphate, while carbons 4 to 6 would become the G3P. Both would retain one of the original phosphate groups, a couple of the original phosphate groups. And only G3P continues directly in glycolysis. So that's why triose phosphate isomerase rapidly interconverts DAP and G3P to ensure that both halves of the glucose is ultimately, processed through the rest of the pathway. So what do you need to remember? Triose phosphate isomerase. Put that in your forehead. Triose phosphate isomerase. Okay, I'm gonna sticky note that. Put it in my ceiling. Triose phosphate isomerase. Word of the day. Triose phosphate isomerase. And we just have another big figure and payoff phase. What's the payoff phase? Glyceraldehyde, the G3P That's where it starts, right? It goes to the payoff phase. G3P is oxidized by glyceraldehyde 3-phosphate dehydrogenase. It uses NAD plus as an electron acceptor, and this oxidation would capture energy by different bonds, specifically the phosphate carboxylic anhydride. Lots of things going on. And then also note we talked about the 1,3 bisphosphoglycerate. No worries. Yeah, thank you for that one. I appreciate it. Just trying to break it down into something that even I could understand as well. So that's why essentially 3 plus 3 equals 6. And 1,3 bisphosphoglycerate can also be converted into 2,3 bisphosphoglycerate. That's just the weird kinases. Usually this isn't mentioned in the figures, but yeah, because normally in the glycolysis pictures you would see 1,3 bisphosphoglycerate into the 3-phosphoglycerate, then 2-phosphoglycerate, then you have the PEP into the pyruvate. But note, between the 1,3 bisphosphoglycerate into the 3-phosphoglycerate, you could have a 2,3 bisphosphoglycerate. This depends on where it comes from. It could come from nowhere. It could just not be there. So ultimately, what is the fate of the pyruvate? We've talked about that a lot. Pyruvate is the end product of glycolysis, right? That's like why we're doing glycolysis. It's from glucose into pyruvate, essentially. It has a lot of different fates. So it's like if you've played an anime game, like an otome game where you have different endings. Pyruvate is the main character. Oh my gosh, this is great. Yeah, Pyruvate is the main character in an otome game. It has different fates. Different fates. Under aerobic, if you have oxygen, pyruvate is transported into the mitochondria and it's converted into acetyl-CoA. Just like note that down. Again, if there is oxygen, so you're pyruvate. If there is oxygen, you're like, okay, I have oxygen. I can go into the route of the TCA or Krebs or citric acid. I will be converted into acetyl-CoA. Who will convert me? Pyruvate dehydrogenase complex. While I'm being converted from pyruvate to the acetyl-CoA, you release CO2 and also something else gets edited from NAD plus into NADH. So again, your pyruvate, you have oxygen, so you can enter the ring, the TCA cycle. As you go inside and become the acetyl-CoA, two stuff is happening. CO2 is released, NADH is also made. So that's what happens. But if you do not have oxygen, if you do not have the key to enter the dungeon, you will, like such as in the muscle or in cells that don't have mitochondria, the pyruvate, would be reduced to lactate. You will be milk. Lactate. Using what? Lactate dehydrogenase. Okay? So again, if you're pyruvate, you just went through glycolysis. You went through a lot of things. You went through, yes, we go with we learn new things every day. Yeah, if you have it depends on if you have oxygen or not. If you are pyruvate and you have oxygen, okay, you can be converted into acetyl-CoA. But if there's no oxygen, you will become lactate. By lactate, dehydrogenase. And this would recycle NADH to NAD+. So it'll change some of the details. Pyruvate can also be transaminated into alanine, just another part. And what does that mean? Well, it is in the nitrogen transport of the body, and links amino acid metabolism with the carbon metabolism. So they just talk. This is like a different dungeon. Now, let's talk a little bit about PFK1, phosphofructokinase. It's a major regulatory enzyme in glycolysis that catalyzes the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. So F6P to F16BP. What is used there? It's PFK1. It sounds cool for some reason. Phosphofructokinase 1. If you just say, I work with PFK1, sounds cool. So PFK1 is inhibited by high levels of ATP and citrate. Why is that important? Well, it's because if you already have a lot of ATP and citrate, you would not need to do glycolysis because you already have a lot of it. I mean, like a lot of the products. So in contrast, if you have a lot of AMP, that would indicate low energy, and that's why you will do glycolysis. You will continue. And if there is also the element of insulin, okay? So fructose 2,6 bisphosphate itself is converted or produced by PFK2. It would convert fructose 6-phosphate to F26 using ATP, and this process is stimulated by insulin. That's just where it happens usually. And just to mini summarize that part, the regulation of glycolysis is at 3 levels. How is it? Why is it at 3 levels? Well, hexokinase has a low Km and low Vmax, meaning it would bind to glucose tightly. I can discuss the low Km and low Vmax in a different mini lecture because that would require talking about the Line-Weaver-Burke plot for inhibition and etc. So that's gonna be another hour. But yeah, so far, just remember, hexokinase has a low Km and low Vmax, meaning it binds to glucose tightly. And then glucokinase is found in the liver and pancreas. It has a high Km and high Vmax. If you go outside the mini box of the DHAP and G3P, you'll find some links to carbohydrate metabolism and lipid synthesis. Those are also important things to remember. You have different oxidation states of carbon as the functional groups shift from alcohol to aldehyde to carboxylic acid. So remember, ethanol is an alcohol, acetaldehyde is an aldehyde, acetic acid is a carboxylic acid. The same as the G3P, it has a fully reduced alcohol. G3 that earlier that was glycerol 3-phosphate. Now we have glyceraldehyde 3-phosphate. It has an aldehyde group. Meanwhile, 3-phosphoglyceric acid contains a carboxylic group. Just to check the the landscape of glycolysis, you have changes in Gibbs free energy, just going from up to down. You have glucose being at a very high energy state. The very first major drop is using hexokinase. ATP is consumed to form G6P. Second major drop is PFK1. And those two steps are highly exergonic, meaning they really want to happen. Exergonic means G minus. Talked about that earlier. And just the overall trend is just downhill. And because oxygen is so much more reactive than carbon, the carbonyl carbon would be a little bit positive. It has a slight positive charge than the carbonyl oxygen, which is a little bit negative. So carbonyl oxygen again is slightly negative because of the oxygen, it's more electronegative. Meanwhile, the normal carbon is slightly positive. Hemiacetals, they are products of the reaction of aldehydes or ketones with alcohols. And with that one, the reaction of ethanol with we have a reaction of ethanol with propanol. Just the oxygen attacking the carbonyl carbon, and boom, you have a methyl hemiacetal of propanol. And let's see. Oh, goody. The next one is the TCA cycle. Okay. So TCA cycle. Should I save this or should I discuss it now? What do I think? Maybe let's just save this one for a mini future thing. All right. Well, that is all for now. Thank you for watching. Bye.